Welcome to the Brain Prize webinar series. We've heard about how neurons and neural circuit activity really robustly influence the growth, invasion, and progression of high-grade gliomas. And today we're going to hear about the way that, in turn, gliomas dramatically affect the function of neural circuits, relevant both to tumor-associated epilepsy as well as patient cognition. And of course, these neural circuit modifications have widespread ramifications as the change in neural circuit activity further augments the growth promoting effects of neuronal activity on the cancer cells.
Yes, also welcome in my name. So Michelle and me, we are really looking forward to really elucidating these complex effects of the nervous of cancer on the nervous system and the vicious cycles that are originating from that, but also of course from a neuroscience perspective. How specifically and also broadly and unexpectedly the nervous system can be influenced by cancer. Today, focusing on the brain. And it's my pleasure to introduce our first speaker, Harald Sontheimer, from the same country than myself, and originally from Germany. Harald Sontheimer is professor and chair of neuroscience at UVA, obtained his master degree in evolutionary comparative neuroscience in '86, and obtained another doctorate in biophysics and cellular molecular neuroscience from Heidelberg, where I'm also from, and today talking to you, and was then moving to the United States for his postdoctoral studies at Yale, and became then independent and moved to the University of Alabama in Birmingham and stayed there for 11 years, from '94 to '15, and then moved to Virginia Tech from '15 to '21. And so Harald has really an amazing oeuvre of work showing the importance of glial support for cells in health and disease. He really elucidated the first mechanisms by which gliomas induce neural circuit hyperexcitability and the associated epilepsy that is so significant and relevant for so many patients with gliomas and other brain tumors. And his work was recently recognized by the 2025 International Prize in Translational Neuroscience from the Gertrude Reemsma Foundation in Hamburg, Germany, actually my home city. So we share a lot in common, Harry, and I'm really looking forward to your talk. The stage is yours.
Well, thank you very much, Frank, for this kind introduction. Let me pull up my slides, share them with you. Laser pointer. Well, thank you both for inviting me for this webinar and allowing me to present the work that we've been doing over the last, over 2 decades. I do want to start out with a case presentation just to frame the topic. This is John, a 38-year-old Birmingham, Alabama school teacher. He was fit as can be, a cyclist, a runner, and one day he woke up with a headache that was unresponsive to ibuprofen and Tylenol. He called it the worst headache of his life. Later in the day, he suffered a seizure. He presented to the emergency room to my colleague, Bert Neighbors. He took the history, which was unremarkable, no history of seizures or TBIs. And he performed a scan on which he saw a worrisome lesion on the right occipital lobe by MRI. And over the next 6 weeks, rescans showed an amazing progression of this lesion to the size of a golf ball. This lesion was partially resected and was diagnosed as a grade 4 glioblastoma. John was treated with radiation therapy and Tumours, which we consider the standard of care. He passed away 18 months later, leaving behind 2 daughters and a wife. And as you'll see in the statistics, glioblastoma are the most common malignant primary tumor in adults. There's about 25,000 new cases in the United States. Peak incidence is later than John's age, over 55, but it does represent a major contributor to cancer mortality in younger adults. Median survival, just as in John's case, about 12 to 15 months. Now, we do believe that these tumors can derive from a variety of cells, but really glia and glial progenitor cells that includes dedifferentiated astrocytes, NG2 cells, but frank undifferentiated type B progenitor cells in the various proliferative zones of the adult CNS, particularly the subventricular zone. Now, just like any other cancer, mutations in tumor suppressor and oncogenes are required for the cancerous transformation of these glia or progenitor cells, and particularly receptor tyrosine kinases such as mutations in the EGFR receptor, p53 mutations, as we'll talk about a little more in this talk, and RB signaling is altered in the majority of these cancers. Now, unlike other cancers in our body, this cancer grows in a completely enclosed space within the skull. So if you look at these 6 presentations of gliomas of various size, various locations, you can see massive expansions of tumors in this example, for example, that can't just be accommodated by pushing tissue aside. If you have a breast cancer, a colon cancer that grows in and against soft tissue, it simply pushes the soft tissue away. But this cancer here has to grow on the expense of the host. It has to eat into the brain and destroy what's there to create room. So over 20 years ago, we came up with the hypothesis, delete this here. We came up with the hypothesis that for these tumors to grow, that they actually have to release a substance, particularly we were somehow or other, that these tumors release glutamate, the neurotransmitter glutamate, and expose surrounding neurons to excitotoxicity. Of course, this was a time when excitotoxicity had been widely discussed in the context of stroke. And I'm sorry. Are we back in live screen? But excitotoxicity has been discussed in the context of stroke, overactivation of NMDA glutamate receptors, or indirect activation of voltage-gated calcium channel, leading to the catastrophic influx of calcium, activating a number of calcium-activated destructive enzymes such as phospholipase A2, nitric oxide, the mitochondrial permeability pore, ultimately culminating in cell death. Now, our earliest finding in support of this hypothesis, or that gave rise to this hypothesis, was as follows. We were looking at the ability of glutamates to be cleared from the medium of cell cultures, and we've spiked a monolayer of astrocytes with 100 micromolar glutamate. And within about 2 hours or so, the astrocytes almost completely depleted this glutamate. We know that we can repeat this again and again. We essentially consider astrocytes glutamate sponges. They very quickly take up glutamate and they convert it to glutamine, the glutamate-glutamine cycle that's been discovered by Leif Hertz in the late 60s for the recycling of glutamate. But what we didn't anticipate is when we do the same experiment, in this instance with the glioma cell line, STTG1, rather than removing glutamate, the gliomas would actually keep adding glutamate to the media at an extraordinary rate, eventually reaching 0.5 millimolar concentrations, 500-fold what we would find typically in the CSF of brain. Now, when we co-cultured neurons, cortical or hippocampal neurons, with gliomas in a way where they shared the same medium but couldn't touch each other, as illustrated here, we found within 24 hours, essentially all of the neurons died. But they could be prevented from dying by blocking the very glutamate receptors implicated in glutamate excitotoxicity using MK801 or CNQX. Later, Peter Hutchinson put microdialysis probes into the brains of tumor-bearing patients and recorded glutamate concentrations rather similar to what we observed in culture and in mice. up to 300 micromolar. Again, 300-fold the concentration of normal, and about 50 times in excess of excitotoxic concentrations. So this is not just a phenomenon that occurs in animals or in cultured cells. It does occur very much in vivo in patients. So how does the glutamate get out of the glioma? Well, years worth of work that I'm going to quickly summarize identified a glutamate cysteine antiporter called System XC as the main culprit. Now, System XC is a transporter that provides L-cysteine as an essential precursor for the synthesis of the cellular antioxidant glutathione. Cancer cells, being rapidly dividing cells, have a lot of mitochondrial activity. These mitochondria create reactive oxygen species. And as a result of that, glutathione is being depleted and needs to be replenished. With every import of cysteine, a molecule of glutamate is being released into a narrow extracellular space, exposing surrounding neurons to potentially excitotoxic concentrations of glutamate. The reason we discovered that this is unequivocally due to this transporter is, first of all, this transporter, the release of glutamate, is not sodium dependent, as is the uptake of glutamate by astrocytes. It is, however, dependent on cysteine. Increasing concentrations of cysteine in a time and dose-dependent fashion drive the release of glutamate. Both glutamine, the precursor for the synthesis of glutamate, and cysteine need to be present for this to occur. In the absence of either or both, it doesn't happen. And we can monitor the increase of intracellular S35 labeled cysteine as a result of the uptake. So unequivocally, this has to be mediated by the System XC transporter, which at the same time had been cloned when we first made these discoveries. And it turns out to be a heteromeric protein that has both a catalytic light chain, that's XCT, and a heavy chain, CD98. The gene for the light chain is SLC7A11. It turns out that this is a transporter that we later discovered, and I'm introducing it now for clarity of the presentation, is inhibited by S4-CBG and other phenylglycine drugs, as well as by sulfasalazine. Now, rather than studying that in cell lines that tend to have genetic drift, we elected to use patient xenolines for all our future studies. By now, we have generated over 100 of these, and these different numbers are just representing different patients. The idea here is you take the tissue from a patient, you never put it in tissue culture, but rather you inject a part of this tumor in the flank of a nude mouse. This mouse will then grow a tumor in its flank, providing it with nutrients and oxygen. And what we observe is that when we implant these tumor cells, they maintain the protein of interest that we're studying in our situation, System XC, very faithfully over many, many passages from mouse to mouse to mouse. What we found is roughly 50 to 60% of patient-derived gliomas very highly express both the catalytic and the regulatory subunit of System XC, as indicated here. The other 30 to 40% have very low or almost undetectable expression. And again, those that have low expression maintain low expression. Those that have high expression maintain high expression in culture. Now, this correlates with high System XC expression with acidious glutamate release that can be inhibited, the sulfasalazine and S4-CPG, versus those with low levels of expression have essentially no detectable glutamate release that can be attributed to System XC and therefore would be blocked by S4-CPG or sulfasalazine. We later discovered that the segregation in whether or not a tumor expresses System XC or not is entirely due to the expression of p53. p53 appears to be a transcriptional regulator. It binds to SLC7A11 gene and it suppresses its expression. So in wild type, we have very low System XC expression, whereas in mutant or in the loss of p53, we have very high expressions of this transporter. Now, to take that a step further, we can show by Western blot here, GBM12 happens to be a p53 null tumor, and you can see very high expression of system XC by Western. GBM14 is a wild type p53, has very low expression of system XC. And GBM22 is a mutant p53, also have very highly expressed system XC. We can use lentiviral expression of p53. In this instance, we take the GBM12 and we lentivirally express p53 wild type. And as you can see, we markedly reduce glutamate release. We can directly regulate expression of this transporter. Now, can we demonstrate in vivo evidence for excitotoxicity as a result of system XC expression? And what you're looking at here is a series of brain slices. We are demarcating the tumor boundary here in a low system XC and high system XC expressing tumor. In the high system XC expressing tumor, the UN label that labels all neurons in that area shows an almost complete absence of neurons, but plenty of reactive astrocytes here shown with GFAP staining in red, compared to the wild type where we have a nice intermingling of neurons and glial cells, essentially a normal brain. We can normalize this loss of neurons or rescue the loss of neurons by treating these animals systemically with the system XC inhibitor sulfasalazine. Now, not surprisingly, high expression of system XC shortens survival in mice by about 50%. So low expressors live about twice as long. And querying the Rembrandt database, we see a similar, more accelerated patient death in those that highly express system XC compared to those that do not. Now, John had an unexplained seizure, and it turns out that about 70% of patients associate seizures as a presenting symptom. Now, in about a third of patients, during the subsequent etiology of their disease, they develop tumor-associated epilepsy. That is, they have spontaneously recurring seizures during that period, and these are often resistant to typical anti-epileptic medications. We are actually able to reproduce this phenotype in mice implanted with the human xenolines. This mouse will just about now show its tail swinging up, will begin to shake, will use its forepass to clean his face. This is a typical sign of a convulsive seizure in this mouse. This is a system XC expressing mouse. In about 80% of these mice, we can observe tumor-associated epilepsy by EEG and by video monitoring. This compares to those that have low system XC where seizures are rarely observed. Now, when we cut brain slices from these animals and we use patch clamp recordings and look at cellular excitability of individual neurons, we can find that in high system XC expressing in animals implanted with high system XC expressing tumors, we see to the same level of stimulation a larger number of action potential discharge to cellular hyperexcitability. We do see a small change in the resting membrane potential. Here are two examples of high system XC expressing tumors. We accelerate seizure generation going into a magnesium-free solution, and then we apply sulfasalazine, and as you can see, we can completely block the ictal activity that's associated typically with behavioral seizures by using sulfasalazine. We're also able to suppress seizure generation in animals that are injected with sulfasalazine. We can reduce the seizure incidence and frequency by about 80%. Unfortunately, sulfasalazine has a half-life of about 80 minutes. So within about 2 to 3 hours, we begin to lose the effectiveness. However, we can retreat these animals, and every time we retreat these animals, we get the seizures under control, suggesting that glutamate release via system XC is necessary for tumor-associated epilepsy. Now, I've been studying epilepsy all my life, and the typical epileptologist is more interested in GABA than in glutamate. It turns out that GABA is a very powerful inhibitor of neuronal activity, and it's hard to overwhelm the inhibitory tone of GABA unless we have a loss of inhibitory neurons or inhibitory tone. So is it possible that the neurons that are being lost are disproportionately inhibitory neurons? Now, to study this question, Banu Tiwari in the lab looked at the excitotoxicity in the vicinity of tumors in tumor-bearing mice. Each of these lines on the left here demarcates a distance of 200 micrometers. He used several markers. First, he used NUN, a pan-specific neuronal marker in red, and as you can see with these snapshots here, there's a reduction in overall neuronal cell number within the first 200 microns of the tumor. We're suspecting that the glutamate has reached these cells and have killed most of those. In green, he stained the inhibitory neurons with the calcium-binding protein parvalbumin. And as you can see here, you have to go, you're losing inhibitory neurons much more completely in the vicinity of the tumor and over a much greater distance. He validated this with a secondary marker, WFA, which stains perineuronal nets, which are found around 90% of PV interneurons. And again, he was able to confirm that, in fact, and the quantification is here, while there is an overall distance-dependent loss of neurons, it disproportionately or more favorably kills inhibitory neurons. But what we also noticed is you look at the structure of these perineuronal nets, and they look very ragged in the vicinity of the tumor. Now, we had not paid much attention in our previous studies to perineuronal nets, but as these two juxtapositions of the contralateral normal brain and the peritumoral brain shows, when we peel back all the other cells, the perineuronal nets are just a ragged shell of themselves, where normally they cover dendrites and the axon initial segment all over the cells. So we got interested in PNNs and wondering whether or not they might contribute to the tumor-associated epilepsy. So Golgi first described those as extracellular coverings. We know today that they were synthesized by hyaluronic acid synthase through link protein attached are neurocans, such as particularly aggrecan, as well as chondroitin sulfate proteoglycans, which are contain very negative charges attached to the CSPGs. They have been shown to be important during the development in stabilizing ocular dominance, for example, in the visual cortex, and they're believed to hold synapses in place. But what we're showing here, for the first time, I believe, by patching onto cells next to tumors that either do or do not have perineuronal nets, is that the presence profoundly affects the firing frequency of these interneurons. The cells that have intact perineuronal nets fire at very high frequency. And again, these inhibitory neurons are known to fire at tens to hundreds of hertz. And of course, with every firing, release the inhibitory neurotransmitter GABA. Those that don't have PNNs fire much slower. Well, if we select cells at the tumor boundary, we fill each cell that we recorded with lucifer yellow, and then we can go back and validate whether a cell did or did not have a PNN. We're able to show that, in fact, next to the tumor, those that have lost the PNNs due to the tumor's presence fire at a much slower rate than those that have an intact PNN. We can phenocopy this change in firing pattern by using an enzyme, chondroitinase ABC, to digest the perineuronal nets. And in fact, that is sufficient to reduce the firing frequency of these neurons. So what Banu did next, he implanted different human-derived glioma lines, and he systematically recorded from peritumoral neurons and did so-called input-output curves. So he increased the stimulus frequency, and he counted the action potentials as a result of that. And as you can see, compared to sham animals, those tumor-bearing animals, these neurons fire at only about half the frequency, and the snapshots are shown here. We did not find very substantive other changes a small change in resting membrane potential, but we found a very profound change in the membrane capacitance of the peritumoral neurons, those that have lost the perineuronal nets. Now, we suspect that the loss of these PNNs were due to the digestion by MMPs, and in fact, we were able to verify that by giving these animals systemically GM6001, a pan-spectrum MMP inhibitor, and we were able to completely rescue the firing phenotype of these neurons next to these tumors. And in fact, we were able to also rescue with GM6001 this increase in membrane capacitance. So what we believe is going on is that the perineuronal nets, owing to their negative charges, act as an insulator. They're thickening the membrane of these neurons, thereby reducing the membrane capacitance. So in the normal state, this permits these inhibitory neurons to fire at supraphysiological rates, at very high firing rates. And again, that drives the release of GABA. The gliomas come along as they grow, they release MMPs, they digest the perineuronal nets. The membrane capacitance increases as the loss of this insulating substance occurs, that reduces inhibitory firing and reduces the GABA release, thereby tipping the excitation inhibition imbalance. Not only is that informative or important to explain the tumor-associated epilepsy, I think we've uncovered a hitherto unrecognized function of perineuronal nets in the normal brain, allowing cells to fire at supraphysiological range, just like the myelin acts as an insulator, reducing membrane capacitance in the axon. The PNNs are insulators of the cell soma. Now, to demonstrate that change in membrane capacitance would be sufficient, we're using a modeling approach where a PD intraneuron here fires at a constant rate, and it inhibits the input of the cortical pyramidal cells here. And in just a few seconds, we will start a countdown. Actually, the countdown has just started. I should probably rewind that if I'm able to do that. Let me start this again. So when I tell you so, when this firing rate goes down, we'll actually change the membrane capacitance. It's right now. Let's see, right here is where we change the membrane capacitance and the cells go into frequent firing mode. We can reproduce the hyperexcitability in these neurons by simply changing the membrane capacitance by the same amount that we've measured in these cells. So we believe that the degradation of the PNNs is absolutely necessary to change the firing frequencies of these PB neurons. The PB neurons fire slower, release less GABA, and thereby tilting the excitation inhibition imbalance. So this gives us therapeutic opportunities. Both MMP could be targeted, as well as the glutamate release from system XE could be targeted. We had earlier mentioned that sulfasalazine, and we discovered that sulfasalazine actually acts as a inhibitor of system XE, and it does so by mimicking the glutamate binding site. So sulfasalazine docks into the glutamate binding site of the transporter, but due to its size, it can't be transported. It's not a good blocker. It has low affinity and it has low permeability into the brain. However, the beauty is it is widely available and it's safe. Patients with Crohn's disease, millions of those patients take up to 1, 2, 3 grams a day of this drug. So we can rapidly use that into, at least in the pilot study, clinically. And that's what we did. We embarked on the biological study, simply using glioma as the glutamate release as a biomarker. We asked whether or not giving a patient for which we have a confirmed status of system XE expression, an acute dose of 1 gram sulfasalazine, and we measure glutamate non-invasively using spectroscopic imaging, using a 3 Tesla magnet, and asking whether within the next 15 to 30 minutes, we can modulate glutamate being detected with this technique, and that was done in collaboration with Bert Neighbors and Adrian Latti at UAB. So here are 4 example patients. Here's the region of interest, peritumoral and tumoral, that we're looking at. For each of these patients, we have from needle biopsies a confirmed status of System XE expression, given here, and the percentage of the tissue being labeled. And as you can see here, in patient 1, almost 70% reduction in glutamate levels in response to sulfazine. Patient 2, lower expression of the transporter, still a 30% reduction. And in the patient where we have essentially undetectable levels of System XE, no response to sulfazolazine. So across the patients we've studied, we see that there is a essentially linear correlation in terms of the decrease of the glutamate signal with regard to SLC7A11 or System XE expression. We did enroll both GBMs and astrocytomas, as well as a couple of oligodendrogliomas, and we found that correlation across the board in these patients. We also validated that patients that expressed System XE and responded to sulfazolazine actually presented to seizures on EEG when we did these recordings. So what I believe we have been able to demonstrate in our studies is that the glioblastomas fall into 2 categories: excitotoxic and non-excitotoxic tumors. The excitotoxic tumors show rapid growth and peritumoral excitotoxicity. They present with high seizure incidence, and they're responsive to sulfazolazine. And we found that they're also mutated in p53, whereas the non-excitotoxic tumors grow more slowly. They show very little peritumoral excitotoxicity and much lower seizure incidence. They're not responsive to sulfazolazine, and they tend to be wild-type p53. Now, the open questions that we're working on right now and that we feel need to be addressed is, what happens to astrocytes? The astrocytes next to tumor become reactive, and do they actually let us down? Are the astrocytes actually to blame? Because it's really hard to overcome their ability to modulate glutamate and remove glutamate. It turns out that when we look at the tumor borders and we look with GFAP, BRDU, and Ki67, we find that there is a large number of proliferative cells. About 6% of these astrocytes in the vicinity of the tumor start to proliferate. We see from the GFAP staining that there is massive morphological changes indicative of reactive gliosis. We also find that there's an upregulation of phosphorylated STAT3, which is often used as a marker for reactive gliosis, but importantly related to glutamate. When we measure the capacity of these astrocytes to remove glutamate, take up glutamate in response to a 200 micromolar glutamate stimulus, we find that there is about an 80% reduction in their ability to remove glutamate from the extracellular space. So they're impaired in controlling glutamate. So indeed, the astrocytes are letting us down. Now, interestingly, in studies that Miguel Escalante does in the lab right now, we find that simply taking a glioma-conditioned media and putting those on astrocytes profoundly changes their morphology. It upregulates pSTAT3, and it actually leads to increased glutamate release, increased expression of System XE, suggesting almost that the tumor releases factors that reprogram the surrounding astrocytes to become more tumor-like and work on behalf of the tumor to release glutamate rather than taking it up. And we're in the process of using transcriptomics to look at comparing directly astrocytes associated with gliomas versus controls. We do see thousands of genes, not surprisingly, changing. That includes those that Shane Littlelow proposed for A1 reactive astrocytes, but there is a very significant number of genes that are unrelated to this canonical change that he has observed in reactive gliosis. And we're trying to decipher what the linchpin is, what are the relevant gene changes that drive this conformation or co-option of surrounding astrocytes. So in summary, we found that gliomas co-opt the neurotransmitter glutamate to actively promote their own growth. That glioma-released glutamate is important for hyperexcitability, but that the hyperexcitability is at least in part due to the preferential loss of PNNs on inhibitory PV neurons, who now slow in their firing and therefore tilt the excitation inhibition imbalance. We do believe that the glioma glutamate transporter SLC711 is a good therapeutic target with some promising data, pilot data in hand. We do need better drugs to target this transporter, but there is the opportunity to disrupt and ameliorate tumor-associated seizure and hopefully stop tumor growth. I am very indebted to funding agencies and a large number of graduate students, postdocs, and collaborators that have contributed over the years to that work, and I'm happy to take any questions.
Beautiful talk, Dr. Sontheimer. Thank you so much. Really, the very first demonstration that gliomas directly influence neurons, and just such an honor to have you here presenting this seminal work. I have one question, and then maybe we can go around briefly. In IDH wild type adult glioblastoma, there's really very heterogeneous regions of the tumor, with the nodular core being the primary place in which neurons, at least glutamatergic neurons, are lost, and then the diffusely infiltrative component, really exhibiting both molecular and cellular heterogeneity, and really having very different behavior, chiefly where the neuron to glioma synapses are forming, et cetera. And so I'm wondering if the system XC expression is different in these different regions of the tumor, and if the perineuronal net loss is also regionally heterogeneous, if it's different in the invading component of the mass compared to that nodular core.
Well, Michelle, those are great questions. We're actually looking at both of those questions right now. With regards to the PNNs, the PNN loss actually is within a relatively narrow region of about 300 to 400 micrometer off the tumor. I do believe that this is a somewhat non-specific effect due to the release of MMPs and other proteases. And of course, tumors, in an effort to invade, release proteases just to loosen up the extracellular space. And the perineuronal net become innocent bystanders, if you will. They will be digested in the process. The MMPs don't distinguish between the interstitial extracellular matrix where there's these compact PNNs. With regards to the system XC expression, we're now using spatial transcriptomics, and that will give us the ability to resolve that, and particularly resolve that in human tissue as well. But this is a great question. There is clearly not a homogeneous expression of this transporter throughout the tumor at all.
Wonderful. Maybe we have time for one more question. Frank, Ben, Sean?
Yeah, thanks a lot, Harry, for this, for you presenting your really seminal pioneer work, introducing this concept of dual activity therapy against epilepsy and glioma alike. In epilepsy, epileptology, we always kind of wonder, are there disease-specific mechanisms that are in place? And of course, you make a strong, your findings make a strong argument. Yes, there are. I just wondered, did you kind of, along these lines, did you check whether these perineuronal nets are really specifically driving epilepsy and hyperexcitability in gliomas, or do you also kind of see these features in other epilepsy models outside glioma? And last but not least, I think sulfasalazine is a great suggestion for this co-treatment. Of course, the alternative would be parenteral, also inhibiting neuronal hyperexcitability and the AMPA receptors on the tumor cell side. So, what are your kind of considerations? What other strategies might actually work or are most successful to, let's say, co-treat both and in the end establish a new pillar of cancer treatment?
Well, funny you should ask, but we actually have been all over epilepsy and perineuronal nets. We've published several papers, and you have a couple more coming. It turns out that perineuronal net disruption is very common in various forms of epilepsies. We looked at pilocarpine-induced epilepsy. We looked at a virus encephalitis-induced epilepsy in the brain. And in fact, what we're seeing here in tumor-associated epilepsy is actually replicated in several other forms of epilepsy. So I would argue that and we've actually had some success in treating with MMP inhibitors, preventing the in vivo loss of these perineuronal nets, and had some success in other epilepsy models as well. So I guess multimodal therapy clearly for glioma patients is definitely warranted in the future, and that includes many things that are different from what we're studying here at it. But it might include targeting the matrix metalloproteinases along with the system XC glutamate release. But of course, I think those are just two components that have to be in a successful treatment. And one also has to look at the temporal evolution of the tumor. At what point in time is the system XC important? And at what point in time do perineuronal nets contribute? And could one maybe treat sequentially and therefore achieve better outcomes?
Wonderful. Thank you so much. This was a wonderful talk, and continuing the theme of glioma influences on neurons and neuronal circuit function, I'm really delighted to introduce Dr. Benjamin Deneen. Dr. Deneen completed his undergraduate studies in genetics at the University of California, Davis, and then focused his graduate studies at UCLA on cancer biology, studying pediatric sarcomas, a topic dear to my heart. And then switched gears in his postdoctoral fellowship, studying developmental neuroscience at California Institute of Technology. In 2009, he started his independent laboratory at Baylor College of Medicine in the Center for Cell and Gene Therapy and Department of Neurosurgery. He also then later founded the Center for Cancer Neuroscience at Baylor in 2021. Over the past 17 years, Dr. Deneen's lab has made seminal contributions to our understanding of glial development, the neuroscience of brain tumors, and glial control of brain circuit function. His work has been recognized by many accolades and includes the 2012 Sontag Distinguished Scientist Award, 2021 DeBakey Award for Excellence in Biomedical Research, 2023 NINDS Outstanding Investigator Award, and the 2024 O'Donnell Award from the Texas Academy of Medicine, Engineering, Science, and Technology. We're really delighted to hear from Dr. Benjamin Deneen.
All right, thank you, Michelle. Let me share my screen here. You guys can see that. Hold on. Coming there. Can you guys see this? Yes? All right. All right. So thank you very much. It's a real honor to be here and sort of share some of my work that complements the seminal discoveries made by Dr. Sontheimer and also Frank and Michelle. So what I'm going to talk about today is understanding sort of how brain tumors and neurons communicate with one another during malignant progression. So no need to sort of beat a dead horse here, but we know that glioma is universally a lethal disease. Survival rates haven't really changed in 60 years. Standard of care hasn't changed in 20. Glioma is comprised of glial-like cells. There's been a big push to understand developmental programs that drive tumorigenesis, as we know that development is often recapitulated during tumorigenesis. More recently, there's been an appreciation by Dr. Sontheimer and Michelle and Frank as well that glial cell function is adopted by glioma cells, and understanding how glial physiology is utilized to promote progression is fundamentally important for understanding diseases and this disease, and for also potentially identifying new cures. So this has led to sort of the field of cancer neuroscience, and this is a wonderful review written by Frank and Michelle and the Cancer Neuroscience team, where basically we show that there's these different types of interactions between tumors and neurons and cells within the microenvironment. There's electrochemical interactions where tumors and neurons make direct synaptic connections. There's paracrine interactions, which is what Dr. Sontheimer was referring to earlier. There are systemic interactions where you might have a tumor in the colon and it could influence reward centers in the brain. Also, breast cancer patients have trouble sleeping. So clearly there are systemic effects that tumors have on the brain. There's interactions between tumor cells, neurons, and immune cells as well. and there's therapeutic also effects. So what I'm going to talk about specifically, and what my work has emphasized, is understanding the paracrine interactions between tumors and neurons. What are the lines of communication between tumors and neurons, and can we decode these lines, and can we break these lines and use them as a cure or potential therapy for these diseases? So again, as I mentioned, tumors talk to neurons, neurons talk to tumors. There's this vicious cycle of cross communication where neuronal hyperactivity drives tumor progression. Tumor progression, as Dr. Sontheimer mentioned, causes release of factors that influence neuronal activity, and you have this self-propagating vicious cycle that promotes progression and network hyperactivity at the same time. So what my lab is interested in, and what I'm going to talk about today specifically, is what are the lines of communication between neurons and tumor cells, and can we identify them, and what does it mean, and can we potentially use this to understand the disease and also to potentially develop more cures for this horrific disease. Okay, so before I get into that, we're going to talk about chickens. So when my lab started many years ago, we were studying sort of chick spinal cord development and early gliogenesis, and we used this wonderful model called the chick model, where basically we would electroporate the central canal of the chicken, and what would happen is all the DNA would go to one side, and we could basically use this as a developmental model to study early gliogenesis and neurogenesis. We published a few papers using this model. And so around, I would say 10 or 12 years ago, about 12 years ago, we became interested in sort of taking these ideas and applying them to brain tumors. So actually, take that back. So here's some of our studies here. We basically show, we use this model to study early gliogenesis, and we show there's all these transcription factors that do things. And so what we really wanted to do was to take this idea of early glial development and apply it to brain tumors. So drawing inspiration from our chick model, we developed this electroporation-based model of glioma. Where what we do is we're able to electroporate the cortical ventricle of an E15.5 mouse embryo. We can introduce piggybacks to drive oncogene expression, or guide RNAs along with Cas9 to knock out tumor suppressors. We can stuff the pups back in the mom. The pups are born. And roughly 2 to 3 months later, they will form this really nice tumor within the mouse. And what's nice about this system is that, number one, a pregnant mouse at Baylor costs about $60. The surgery takes about 30 minutes. Each mouse has 8 to 12 embryos and produces about 6 to 10 tumor-bearing mice. Importantly, has an intact immune system, and also it occurs in a native autonomous manner. So it allows us to study normal tumor growth in the context of an immune system. So this was a big advantage. And so we initially were going to use this to study early developmental events and how they promote tumor growth. However, as we all know, when you build a new tool, it often creates new opportunities. So this is where we kind of dug into using the system to understand the functional genomics of glioma. And so we know that over the last 20 years, we've done a great job of cataloging all the mutations, or a lot of the mutations that are present in cancer, to the point where we're close to saturation mutagenesis. I think there are roughly 3 million cancer-associated mutations present in the COSMIC database, yet we only know what 5,000 of those mutations do. Here we have 3 of the key tumor suppressors that are mutated in glioblastoma. And you can see on the lollipop plot, there are a host of variants associated with P53, P10, or NF1. So we know that each gene has a host of variants, but we don't know what those variants do. We don't know if those variants are drivers or if those variants are passengers. And so decoding driver variants from passenger variants is a really important aspect of functional genomics of cancer, because it will tell us more about the disease, and it will tell us which variants are important, which ones are not. So in GBM, we sort of focused on EGFR and PIK3CA. These are 2 of the most commonly mutated oncogenes found in the disease. And so the idea would be to take an allelic series of EGFR and PIK3CA and ask which of these variants are drivers, which ones are passengers. To do this, we developed a barcoded system using piggyback transgenesis in our IUE system, and the idea would be to put an individual variant in the piggyback next to a unique barcode, then introduce a library of variants into a mouse. When a tumor forms, we could then rip out the tumors, sequence for barcodes, and use barcode enrichment to indicate whether a variant associated with that barcode was a strong driver or a weak driver. So that was the system that we set up. And the idea, again, was to screen for EGFR and PIK3CA driver variants in glioblastoma. So we spent a year developing the system, executed the experiment, and it worked beautifully. So here's one of the examples. We took PIK3CA, we put in 27 variants, we did our barcode sequence. We basically, when a tumor formed, we ripped out the tumors, we did barcode sequencing, and you can see here with the red bars, you can see we had about 4 variants that were very strong drivers. We then went back and we individually validated each of the drivers that hit, and you can see here, basically, long story short, those that hit, the barcodes that were enriched in the pooled screen individually validated in terms of having decreased overall survival. And I'll add that some of these survival curves have over 40 mice, speaking to how robust our mouse model is. Did the same thing with EGFR. Again, the red bars indicate the variants that have barcodes that were enriched. Individual validation more or less kind of proved the same thing. So we spent a year doing the screen. We had the data. We did not know what to do with it because I was sort of like, who cares about these variants? It doesn't mean anything. What are we going to do? So the next thing we did was focusing on PIK3CA. We sequenced the tumor-driven variants. We found something very interesting. I expected each of the variants to behave very similarly, which is what you kind of see here with M1043I and R88Q. Have this group of tumor-driven variants, that basically behave the same. However, C420R and H1047 behaved very differently from that core group and from one another. So this was very striking, that tumors driven by variants that differ by a single amino acid were different from one another and different from the expected core group. We looked in the gene ontologies, and it turns out that C420R and H1047R had very different expression of synaptic constituencies. C420R had a massive downregulation of synaptic genes. H1047 had a massive upregulation of them. So as Dr. Sontheimer mentioned, this idea of synaptic imbalance, right, this EI balance is sort of associated with epilepsy, and the fact that these variants had this dysregulation of synaptic gene expression got us thinking about synaptic imbalance. And we know from some of our prior studies that our mouse model has robust and reliable presentation of seizures. So we know that this model, if we do EEGs over time, we know roughly 2.5, 3 months in, these mice are going to get seizures. Also, if we look more carefully, we can see that there's this enhanced synapse formation sort of interwoven with the tumor, where you can see there's these synapses right next to the tumor, again, suggesting that there's some kind of synaptic imbalance or a promotion of synaptogenesis in the presence of these tumors. So this led to sort of a very basic question, that is, do the PIK3CA variant-driven tumors differentially initiate network hyperexcitability? Can these variants differentially promote glioma-related epilepsy, glioma-related seizures? So to do this, we generated mice that overexpress these different variants. We used R88Q as sort of our base control model. Here we have C420R and H1047R. We took these mice, we hooked them up to electrodes. We measured spikes, we measured seizures, and as you can see here, both H1047R and C420R had a very early onset of frank seizure presentation. They also exhibited very early spiking, suggesting that there was widespread network hyperactivity that was induced by tumors that express C420R or H1047R. We also did this in PDX models as well. So the next thing we want to know is, how are tumors promoting this network hyperactivity? So we know that astrocytes, again, going back to glial biology and physiology, we know that astrocytes can release a lot of things, including glutamate. They also release factors like transpondins and glycans, and these factors are released by astrocytes. They travel to synapses, and they actually engender synapse formation. And what we found is a member of the glycan family, glycan 3, is highly expressed in C420R and H1047R expressing tumors. So the next thing we did was to overexpress glycan 3 in our model, and we found that overexpression of glycan 3 led to a decrease in survival and a very early onset of network hyperactivity, along with frank seizure presentation. So we think what's happening here is that the tumors are releasing glycan 3, it travels to the local synapses and sort of engender synapse formation towards hyperactivity. So to summarize this work, what we basically use, again, is a beautiful story of serendipity, where we use functional genomics to understand the neuroscience of brain tumors. We created this discovery platform in a native model. We found that tumors driven by related PIK3CA variants have distinct transcriptomes. These variants can selectively manipulate the neural microenvironment to remodel synapses towards hyperactivity. And we found glypican-3 is one of the key molecules. And I want to add that we collaborated with Jeff Nobles, Ken Scott, Asante, and Kawana were the ones who really did all the work. So that's using functional genomics. So now that we have this model, we didn't sort of turn the page to the other question, and we wanted to know how do neurons better understand how neurons communicate with tumors. And again, what we really took advantage of in this model is this infiltration. So here we have our in utero electroporation model. Here you can see a tumor in one hemisphere, and you see this really nice infiltration along the white matter tract to the contralateral hemisphere. We wanted to understand more about this process. And so a lot of the seminal work done by Michelle has shown that if you activate neurons adjacent to a tumor, you will get enhanced growth. So my student Emmett, he wondered what happens if you activate neurons far away in a remote location. So the experiment that he performed was he generated an IUV tumor in one hemisphere, then he activated neurons in the contralateral hemisphere. I thought this was insane. There's no way this was going to work. So the experiment was basically generate an IUV tumor, inject a dread-based tool that will allow us to manipulate neuronal activity in the contralateral hemisphere, treat the mice with the CNO drug, which basically activates the dread and activates neurons, and then monitor infiltration. And in this story, I think in this case, a picture is worth 1,000 words. Here we have the saline control, viruses, no neuronal activation, the CNO, which stimulates the dread and stimulates neurons. You can see that this tumor grows a lot faster. And also you have this sort of infiltration towards the site of activity. It's almost as if the tumor is moving like a moth to the flame. It's attracted to this activity in some way. This is a very striking observation, and it got us wondering, sort of more broadly, can we use this phenomena to study glioma infiltration? Because as we all know, a surgeon can go in and take out the tumor core, but it's those micro leading edge pieces that remain, the infiltrating pieces, that really sort of cause recurrence and are often responsible for morbidity. So to study this phenomena in the context of infiltration, we did single cell sequencing. And yes, I'm aware that the immune system is different. More on that in a month in Spain. However, what we really wanted to study here were the tumor cells. And you can see that if we zoom in on just the GFP positive tumor cells, you can see in the presence of neuronal activity, you have this purple population that's highlighted by the red arrow that is not really present in the control. So this we hypothesized to be the activity-dependent infiltrating population. We did spatial transcriptomics, and turns out that the molecular signature associated with this population is enriched in regions R6, R7, R8, which represent the secondary site or the leading edge. So we think that this activity-dependent population that we identified here is in fact this sort of activity-dependent invasive leading edge. To understand more about this population, we dug into the gene ontologies, and it turns out one of the things that really popped to us was this gene was enriched in axon guidance genes. We know from development, again, everything comes back to development, that axon guidance molecules play a key role in chemotaxis and cell motility during development. So we sort of zoomed in on these axon guidance genes, did a very complicated barcoded screen, which I will not describe, but we basically functionalized this data set using this barcoded screen. I guess I will describe it. Sorry about that. So what we did was took all 43 axon guidance genes, put a barcode on them, electroporated it, and looked basically for the barcodes that were enriched in secondary sites. We compared secondary to primary sites. What we found was a host of genes that were enriched in axon guidance genes that were enriched in secondary sites, suggesting that they were driving infiltration. One of these genes that we studied was CEMA4F. Here we have the survival data. Overexpression, you get decreased survival. Knockout, you get a slight change. You get a slight extension of survival. But clearly, when we overexpress this gene, you have this, the tumor doesn't even grow as a ball. It just sort of becomes this massive, moving, like really spread out invasive tumor. So the next thing we did was once we sort of define the phenotype of the CMO4F overexpression tumors. We use single cell sequencing. And again, what we found here was a lot like our CNO group, with our activity group. We found that the gain of function tumors were also enriched for this prospective leading edge activity dependent population. We dug a little further into the CMO4F gene ontologies, and it looked like there was all of these synaptic genes that were sort of deregulated when CMO4F was overexpressed. So we then asked the question, how does this CMO4F drive glioma infiltration? We looked at synapses at the peritumoral margin and the gain of function, and found that there was an increase in excitatory synapses, a decrease in inhibitory synapses, getting back to what Dr. Sontheimer said about excitatory and inhibitory imbalance. We did some EEGs on these mice, and turns out that the gain of function of CMO4F also promotes network hyperactivity and early onset of seizures. So to summarize this work, what we basically found is that these cortical projection neurons and their activity seems to drive infiltration like a moth to the flame. We identified an activity-dependent infiltrating population. CMO4F seems to drive infiltration, but it also seems to remodel synapses towards hyperactivity, and its role in normal brain has been shown to actually influence synaptic function as well. Okay, so that's sort of one story. So next, shifting gears a little bit, we know from Mario Suba's work and a lot of other people's work that there's a lot of heterogeneity, and we know that there's these NPC-like cells, OPC-like cells, mesenchymal-like cells, but no one talks about the astrocyte-like cells, right? And Peter Dirks basically showed the same thing, that you have these stem cell things, and you have like this astrocyte-like cell type, right? We know the OPCs are prospective cells of origin. We know NPCs have long been implicated in GBM, and we know that the mesenchymal-like and immune cells have emerged as a key component of progression and therapeutic targeting. But what about the astrocyte cells? No one talks about astrocytes and how their biology might be co-opted within the tumor to promote tumor growth. So we know that, as Dr. Sontheimer mentioned, we know that astrocytes have a complex morphology. They are closely aligned with neurons. Here they are in the normal brain next to a neuron. Take a slice through an astrocyte. They have all these processes and branches. At the EM level, you can see they sit right at the synapse. So they're closely aligned with neurons, and these functions might be reutilized in tumors. Recently, our lab basically published a paper where we showed that astrocytes gate serotonin through an epigenetic intermediary during olfactory sensory processing. What we basically showed is that there's a serotonin transporter expressed in astrocytes that allows serotonin to come into the astrocyte, where it then travels to the nucleus, where serotonin is added to histones, and it behaves as the epigenetic modification on histones in astrocytes. And so we wondered, is this mechanism also present in different forms of brain tumors? So can we apply this concept of this sort of astrocytic mechanism to a brain tumor? And so this leads me to ependymoma, which is a form of pediatric cancer. This is in collaboration with Steve Mack's lab. This is our joint student, Eileen. So there's different forms of ependymoma. I'm not going to get into it, but basically we're focused on the relay fusion supratentorial ependymoma. It's thought to be derived from glial progenitors, has a 10-year survival of roughly 50%. However, these tumors are notoriously chemo-resistant, and surgery and radiation are the only interventions. And it's also been shown that epigenetic mechanisms are key drivers of this disease. Our lab, in collaboration with Steve's lab, developed an IUE model of ependymoma, where we can basically overexpress the fusion and knock out P53, and we get tumors that at the molecular level resemble the relay fusion supratentorial ependymoma. So using this model in human diseases, we looked very closely, and what we found is that patients that have the relay fusion, ependymoma patients with the relay fusion, have an enrichment for monoamine transporters and a lot of these neurotransmitter transporters, more so than non-fusion bearing ependymoma patients. We went to our model and we validated expression of these transporters in human and in our mouse model. So we know that the monoamine transporters are expressed in relay fusion positive, or rather they're enriched in relay positive fusion ependymoma. So this raises the question, getting back to serotonin, how does this relate to serotonergic neurons? So before we can do that, we first wanted to know, does activity influence ependymoma? Because we know it influences DIPG, and we know it influences adult glioma, but what about ependymoma? So basically in this experiment, we kind of repeated the same experiment, but we activated neurons on the ipsilateral side, and we stimulated them with the dread for 10 days, with the BRDU. And in fact, when we stimulate neurons, cortical neurons, next to a relay fusion ependymoma, we get this really nice expansion of growth and proliferation. So what happens to the activity of, if we manipulate the activity of serotonergic neurons? So in this model, what we did was we generated an IUE tumor in the cortex. We then manipulated, we activated or suppressed neurons in the dorsal raphe nucleus, which is basically where the serotonergic neurons that project to the cortex reside. So we're basically inhibiting or activating the serotonergic neurons. And what we found is that when we activate the dorsal raphe nucleus, the serotonergic neurons in that population, what we found is there is a decrease in proliferation. So stimulating these serotonergic neurons seems to suppress ependymomal growth. When we suppress serotonergic neuron activity, what we found was an increase in growth. So this is a little bit different, right? It's not a one size fits all. It's not just pan-neuronal activation promotes growth. Here we have a circuit-specific effect where activating a specific set of neurons actually gives you less growth, and inhibiting those neurons gives you more growth. So there's clearly circuit-specific effects on tumor growth, and it could also depend upon the subtype of tumor as well. So given this observation, we next asked, what about histone serotinylation in ependymoma? So we got the antibodies, we stained, and clearly within human tumors and our mouse models, you can see that there is really nice histone serotinylated staining in mouse and human tumors. And you can see in our ependymoma tumor, you have a really nice Western blot here showing this massive increase in histone serotinylated levels of histone serotinylation in the ependymoma tumors. Next thing we did was we collaborated with Ian Mays' lab, who actually discovered the histone serotinylated mark. He gave us a dominant negative form of this histone. We overexpress, so this dominant negative is incapable of having the serotinylated mark added to the histone. We overexpressed this dominant negative, and we got a nearly complete block in tumor growth. So this tells us that on one hand, serotonin seems to suppress ependymoma tumorigenesis, but the mark itself seems to be important for tumor growth. So given this, the next question we asked is, what are the patterns of histone serotinylation within the genome? So here we did CHIP sequencing, and basically we found a couple of things that are really interesting. Number one, we found that it seems that this histone serotinylated mark seems to sort of straddle open chromatin states, where you have open chromatin, and right next to the open chromatin states, you have this histone serotinylated mark. The other thing that's interesting is that this histone serotinylated mark is also right next to where the relay fusion oncofusion binds. So here's a classic ependymoma gene called CCND1. Here you have bonding of the relay fusion, you have an open chromatin state, and you have it sort of straddling straddled by these histone serotinylated marks. So next we did a bunch of transcription factor analysis, and we found a bunch of ETS-related proteins that are involved, one of which is ETV5. I won't get into it, it's a whole other part of the story, but to sort of summarize what we found here, we found that histone serotinylation regulates ependymoma tumorigenesis through these neurodevelopmental pathways. The activity of serotonergic neurons suppresses tumor growth. So again, this could be an ependymoma-specific phenomenon, and it really speaks to circuit-specific and tumor-specific relationships with neurons. We found that basically serotinylation intersects with this relay fusion. What I didn't talk about is the ETS protein ETD5 regulates tumorigenesis, and also neuropeptide Y appears to be involved in suppressing brain network hyperactivity, and that ETV5 seems to suppress a suppressor of network hyperactivity in this model. Okay, so the last vignette I'm going to share in my talk today is about patch sequencing. So we're going back to adult glioma. So for those of you that don't know, patch sequencing is a tool that's used in neuroscience. It's a very powerful tool. Basically, what it gives you 3 levels of information. You can basically stick an electrode into a neuron, and you can record its physiological activity. Then you can use that electrode to suck out the nucleus, so you can use single-cell sequencing to get the molecular properties. And then you can dye fill that neuron, and you can get morphological properties. So it's a very powerful tool for aligning physiological, molecular, and morphological information of neurons, or really any kind of cell. So in collaboration with Ganesh Rao, my student Rachel Curry sort of cooked up this idea of doing patch sequencing of human glioma tumors in collaboration with Xiaolong Zhang, who is sort of one of the co-discoverers of patch sequencing. So the idea here would be to take a freshly resected human tumor and within 90 minutes have it sliced and on a block where we can then do physiology over the course of hours in CSF and then take out the nuclei when we're done and then dye fill it. So we were able to, this is an amazing workflow that we were able to develop. And what we found is what's really interesting. We found that there were tumor cells that fired one single action potential. And the way that we know that these are tumor cells is retrospectively, we're able to go back with the sequencing data and use copy number variants and other criteria to define tumor and non-tumor. So we're very, very sure that these are in fact tumor cells that are firing a single action potential. And so if you look closely at the morphology, you sort of have this really odd morphology where they're sort of glial-like, but then also they have these weird dendrite or axon-like projections. So we sort of term them hybrid cells. So what are these cells and what are they doing? So this is where the molecular information comes in. So here we have these are all the cells that fire that have this weird identity. But what we basically found is that the cells that fire single action potentials have GABA OPC-like properties. They have OPC, they look like they have molecular features of OPCs, and they also express a lot of the GABA channels and receptors consistent with sort of this inhibitory neuron type phenotype. So next we wanted to know, can we validate this and what does it mean? And so we're able to validate the fact that a lot of these IDH mutant, this is all on IDH mutant tumors, that these IDH mutant tumors in fact express that these IDH mutant tumor cells express ALK2, which is an OPC marker, and also express a lot of these GABA channels and GABA associated receptors. What was interesting though is we found that if we go back in patient data, we found that patients that have a high degree of this GABA OPC signature that's associated with firing of action potentials tend to have a much better survival than those that don't have it. So it seems, again, we don't know for sure, but it seems as though these patients that have these cells or more of these cells that fire action potentials seem to do a little bit better. And so this is sort of the summary of that work where we did patch sequencing. We had half the paper sort of describes this SCRAM technology or this SCRAM algorithm that we developed to analyze this data. But the idea here is that we found these hybrid cells that contain spiking GABA OPC-like cells within the tumor. And sort of summarize what we found here, we talked about the lines of communication. We talked about PIK3CA variants, sort of differentially driving network hyperactivity. Glypican-3 as a key factor released by tumors that promotes network hyperactivity. Cortical projection neurons on the contralateral side, sort of like a moth to the flame, driving infiltration, in part through SEMA4F. We also talked about dorsal radionuclear neurons, serotonergic neurons, histone serotinylation, and neuropeptide Y as important factors in sort of mediating tumor neuron interactions. And this sort of aligns with the work from Michelle's lab, where she showed neuroligin-3, ADAM10 are important neural cues, and also tumor neuron synapses are important. And we sort of showed that now tumor cells can also fire action potentials. So with that, I will thank my lab. They're a wonderful group, and this is our team. And thank you very much. This has been a really fun webinar, and I'm really happy to share my work with you guys. Thank you.
Yeah, thank you so much, Ben. This was a really an amazing tour de force through what you have done to Cancer Neuroscience and what you have contributed to the field, really from genomically hardwired ways how tumor cells can influence neurons, including epileptogenesis, to neurotransmitters binding to histones, and then also kind of creating very plausible oncogenic effects. That's really amazing, really kind of breakthrough concepts, I would say. Maybe I would like maybe to start our discussion with focusing on this last part. And you have probably seen Leanne Lee has also shown this similar things for small cell lung cancer. And it seems now, so in a collaboration, it seems we are seeing action potentials also, and others are seeing action potentials in glioblastoma cells, even from wild-type, IDH wild-type glioblastomas. So it could be, so we have never seen those in the animal models so far, and we have also missed these very neuronal glioma cells in this animal model. So what is your take on that? Do you also think that we should study more and more in kind of human material, organoids, tumoroids derived from humans? What is your idea? What are these single, pretty, I would say, low amplitude action potentials doing in these cells? Is it possible that the tumor cells are really catching up? So when there's not enough neuronal input in this specific area or tumor cell or whatever, or disease subtype, the tumor itself is then kind of creating a situation where it kind of stimulate itself action potentials, or is it rather GABA inhibitory? What happens there? So what are your ideas on all these questions? Sorry,
that's a lot of questions. It's a lot of questions. So I'll start with so we've done this in our animal model, in our IUV model, and we do see a similar phenomena in our animal model. So I'll say that we could use the animal model to study part of this, but this is a very difficult question to answer, right? Because I don't know that the organoids are going to really give us the right answer or give us much insight. The human studies are very difficult because of the access to tissue and it's very idiosyncratic doing this experiment. So I think a better question. So I think moving forward, we can use our animal model or even PDXs. I'm sure that we'll be able to see it there, right? But I think more importantly, what does it mean, right? You have maybe in the IDH mutant tumors we saw maybe 30% of the cells have this ability. What does it mean, right? Like what information are they releasing out into the microenvironment that is influencing neuron? We don't really know, right? So part of me thinks if you tie it back to our moth to the flame phenomena, right, where they move from one side to the other, that I think that there's some kind of ion attractant. I think that dynamic changes in ions, because along the white matter nodes of Monje, you have a massive exchange of sodium and potassium. And I wonder if there's something about ions, potassium, sodium, whatever, that serves as some kind of an attractant for these cells, and that perhaps when they fire a single action potential, that's them sort of rewiring the microenvironment to make it more permissive or maybe less permissive, don't know, for tumor expansion and infiltration. So that's my thought today, but that could change because this is all very new stuff, right? And I mean, we're just learning as you are.
So you're following up on this, on the downstream effect, so to say, of this action.
Maybe.
Okay.
Of
Sounds
course.
good.
I'll talk to you in Spain about it.
Oh yeah, great. Over a tequila or something else. Yeah.
Yeah, sure.
Looking forward to that. Or a glass of red wine. So Harry.
Yeah. So Ben, this is really exciting work, and we had a similar observation back in '98. I was working with Dennis Spencer at Yale. He's a neurosurgeon, and he gave me access to human tissue on a daily basis. And we published a paper showing action potentials, particularly in lower grade astrocytomas, pilocytic astrocytomas. They would not fire spontaneous action potentials because their input resistance was relatively low, but we could drive them to fire action potentials. So I wonder, and of course, then came Dwight Bergel's work showing action potential-like responses in sodium channels and OPCs. I wonder whether the sodium channel expression is more of a biomarker for an immature state of cell, and whether or not, just like you and I believe that the tumor co-opts astrocytes to become tumor-like, that they might, as they migrate along the white matter, do the same thing to oligodendrocytes, actually coax them to adopt a more immature state and therefore look a lot more like an OPC cell electrophysiologically.
Yeah, no, that makes complete sense. That firing may in fact not be reminiscent of neuronal firing, but more of an OPC type of firing dynamic. Could totally be that. Right? It'd be interesting to see if again, we couldn't do spatial on these cells, right? Because it was a very precious sample. But it'd be interesting to see if this GABA OPC signature is in fact at the leading edge, right? Or along white matter tracks in some way.
The one other idea, I've been studying sodium channels in glia for 10 years or so, and at some point got really frustrated that we never could figure out what to do. But the best we came up with is that it's a return pathway for sodium ions, into the cell. And a lot of transport systems are actually using sodium gradients, for the transport. And we actually looked at maybe the expression of sodium channels actually as a return partner. It's just to balance the ionic gradients across the membrane. And, whether that's true or not, we had some evidence for that in astrocytes, but it's really an exciting finding.
Yeah, it could very well be. So we're definitely digging deeper. Thank you, Harry. I appreciate it.
But it's certainly an also amazing addition to this kind of aspect of tumor cell autonomous neural features, that we really probably need to look into brain tumors, but as Leanne Lee has shown, also extracranial cancers increasingly now. It's really amazing. So looking forward to particularly to the biological effects of those recordings. So I think for the sake of time, we probably need to move on and come to the last presenter. That's Professor Sean Harvey-Jumper, who holds the Mitchell-Berger Endowed Professorship in Neurosurgery at the University of California, San Francisco. Is also a Vice Chair of Education there, and he's really a prototypical neurosurgeon scientist who contributed tremendously to the question, how can we use refined intra-perioperative recording techniques to understand these bidirectional neurocancer interactions. This is true for low and high grade gliomas and within the functional brain regions, and by these really physiological mapping technologies, I think it really showed very convincingly that this is not just something that happens in our animal models, but is really operational in the human disease and drives also prognosis there for these tumor patients. So Sean also heavily focuses on mechanisms that can be targeted, so translation into new therapies, including neurotransmitters and antiepileptics that really can influence these neurocancer interactions in patients. So Sean, we are very much looking forward to your talk and what you can tell us about the real thing, the cancer patient.
Thank you so much. This has been an incredible morning or afternoon, wherever we are in the world. I've really enjoyed Professor Deneen and Sontheimer's talks. Again, I'm Sean Harvey-Jumper, and thank you for that kind introduction. I'm going to take a slightly different approach in terms of the premise. I'm coming at this, as a functionally active, clinically active neurosurgeon scientist, and there are a couple of weird, interesting phenomenon that brought me into this space. And first and foremost is the fact that many of our patients, as we know, pass away and have a tough time with disease, with cancers and cancer progressions, but there are increasing numbers of people and patients who are living with this diagnosis. And this is particularly true for patients with IDH mutant tumors. The number of cancer survivors is increasing throughout the world. It's not perfect. Glioblastoma remains a really tough disease to treat, but we are getting more and more treatments. So this idea of survivorship is actually what brought me into this space. The other aspect of this that started to make my lab question and think about these sort of experiments that we'll talk about today is really focus on these studies. So on the left side, we have a survival analysis. This is recursive partitioning analysis for patients with IDH mutant gliomas. And on the right side, IDH wild-type glioblastomas. And in each of these, we spend a lot of time talking about folks on the far left side, right, of these Kaplan-Meier survival curves, the worst prognostic outcome patients, folks who do the worst. But the truth of the matter is, there are many patients who will live a reasonable amount of time with significant disease burden in areas of the brain that we can't remove, right? And so when we think about in the human brain, well, what does glioma neuronal interactions or cancer neuron interactions matter? Um, Not only is it the disease that's maybe, less evident and not visible by imaging, but many times we leave tumor regions behind. And in about a third of patients with IDH mutant gliomas, and also roughly a third of patients with glioblastoma, there's significant residual there. And what does that brain do? And that's really what got me into this. And I take even the setting of our best targeted molecular therapies, for example, on the left side of the screen, it's a famous Indigo trial now, folks who are now on IDH inhibitors. But we know that these therapies, although effective, do not shrink away the tumor. The tumors don't go away. They have residual tumor that's evident. And so this idea, whether it's a 57-year-old engineer who will have tumor remaining in the primary motor cortex, thalamic tumor left and head of caudate for a diffuse midline glioma, or patients, again, a IDH mutant patient in their 40s, works as also an engineer, programmer, it's going to have disease left in the ventral thalamus, etc. So that's, again, what got me into this idea of glioma neuron crosstalk and this idea that both excitatory and inhibitory cells have a significant role or impact on cancer initiation, tumor growth, progression, cell invasion, treatment resistance. We've all heard these concepts. We also know that malignant cells, both IDH wild type and mutant, now known, have impacts on neurons and neuronal circuits, both with respect to cognition, but also with respect to seizures. That's some of the beautiful work we saw from the Sontheimer group. In our field, we have amazing tools that have been advanced. This is an example of a calcium imaging of the glioma neuron co-culture condition, and we can see, again, the rhythmic activity of both tumor-associated neurons as well as malignant cells in this glioblastoma co-culture. In my lab, we wanted to try to see if we could move past this and think about what this might look like in the setting of patients. And to do that, we had to think about ways of recording this activity, both, again, invasively and non-invasively. We'll talk about invasive recordings. We started off in my lab using standard of care under clinical context data, and is really focused on subdural low and high density array data for glioma infiltrated cortex. And these are patients who are undergoing a standard of care clinical craniotomy. This is an awake craniotomy, as many of you know. And in that context, patients, again, are awake and behaving, and prior to tumor removal, they perform a series of tasks. What we're seeing on the screen left is simply a semantic naming task. Patient sees an object, and they see that object and both auditory and visual. And you have activation of the dorsolateral prefrontal cortex and then primary motor cortex. And then we record the neuronal activity. And I think of this as ensemble of activity from tumor-associated neurons as well as depolarizing malignant cells potentially. We don't know the separation between the two. We have some work under development in the lab focused on single unit physiology, but I won't present that today since that's still under review. We've started off really in collaboration with the Monje lab. Early on, this is Michelle and Hamza Venkatesh's 2019 paper, in which they were going through reviews, looking at glioma to neuron synapses, and they were asked for human correlation data. And this was when we were beginning the process of analyzing these sort of analysis. And we said, let's take a look at subdural array data during resting state. That means a patient is in the operating room. They are awake with anesthesia off for a requisite amount of time. They have a behavioral task that is called a wakefulness task we administer, in which they are, again, eyes open and engaging, and they perform a series of button presses to assign where boxes on the screen and count aloud, and basic behavioral tasks, just to compare that with their baseline pre-surgical performance on that task. And once they hit, or if they hit a certain threshold, then we use their recordings. And you can see here again, using high gamma power This is subdural high density array, or this is actually low density array at this point. Local field potentials within the high gamma region and the high gamma activity. We're focused on tumor infiltrated regions of brain. These are that infiltrative flare region, the tumor core, that's the necrotic regions, and then healthy appearing regions in which we don't see any evidence radiographically of tumor infiltration. And we can see, again, high gamma power being a proxy of neuronal activity, that there is an increase in high gamma power neuronal activity across and within subjects, which at resting state let us know or gave the suggestion that even at rest, there might be this increase in activity that correlates with some of the preclinical findings or observations that have been published or we're going through in the field. So that was really encouraging for my group. And so we started to think about, well, what can we do or think about more with respect to task-specific behavior? And again, under clinical context at this early stage, task-specific behavior really meant patients undergoing a series of speech, language, and cognitive behavioral tasks. And initially, my thought was to actually use non-tumor infiltrated electrodes, meaning regions of brain that are not annotated as a tumor involved based on flare signal, and use those electrodes to recapitulate what we think about speech outcomes or outputs in the normal setting. So in that setting, we'll say here, this is purple is the electrode coverage over the perisylvian language network. We can see, again, speech onset is time point 0. And if we ask, again, show a subject or participant a individual task, and we break each of these up by millisecond into trial by trial or object by object, 800 milliseconds, 600 milliseconds prior to speech onset, we should see an activation in the dorsolateral prefrontal cortex in that region in normal appearing electrodes, and we do. And then as the person begins to speak, you say the word cat, the motor larynx and pharynx shows an increased activity, which we can see. Then that region begins to suppress as you load up for the next object. So this is, again, cancer patients, normal appearing electrodes, showing that you have a fairly normal circuit dynamics for a speech initiation or lexical retrieval task, which is, again, no surprise and fun to see. But we decided to, based on the work of, again, Sontheimer, Deneen, Monje, Frank Winkler, and others, start to say, let's look at this tumor-infiltrated cortex, assuming that these regions will be wildly disorganized, not relevant, and this would be a kind of a one-time throwaway experiment, just that we would run, for fun, for kicks. And we can see, again, now looking only at tumor electrodes across subject analysis, 1,800 milliseconds prior to speech onset, we still see a temporal pattern of speech activity prior to speech initiation, as you would expect within a normal condition. And then at time point 0, when the person begins to speak, motor larynx and pharynx activates with a post-naming suppression similar to normal patterns. That was quite interesting and actually really surprising observation, because now we're saying speech initiation recruits a tumor language network area that was actually considered until that point disorganized. And so the fact that you have normal organization or a normal appearing organization of speech circuits made us look into this further. And he said, all right, let's separate our electrodes into normal appearing and tumor infiltrated. And let's measure our high gamma power in each of those conditions, both across the time series and in a time series dependent manner, with the dash mark in the middle being when the subject begins to speak. And not only, again, black line shows us our normal appearing regions, but our tumor infiltrated cortex was not only, again, giving us task-specific responses in a normal pattern, but it was hyperexcitable relative to normal conditions. So that, again, didn't quite believe it, actually. I said, there must be something wrong. So let's throw the system into overdrive. The first set of analysis was really agnostic to type of words. And so there's a couple ways, if you can think about it, putting a speech initiation task like this into overdrive. You can gauge the prompts, stimuli that you give a patient based on poly versus monosyllabic or low versus high frequency, we can make the system harder or easier, still under clinical context. So we ran the same thing, separating out monosyllabic versus polysyllabic words. Again, the computational architecture in the brain, you'd imagine to say the word aardvark is different from saying the word cat or harder. We align our ERSPs, or I'm sorry, align our speech responses and our electrodes, generate ERSPs, and separate out and determine if we can assign classifiers to distinguish between the two conditions. So first thing we identified, again, that speech representations in cancer-infiltrated brain does not have the distinct or discrete dorsolateral prefrontal cortex activation you would expect for speech initiation. It was a broader area represented throughout the Perisylvian language area in the dorsal and in the frontal lobe. Second, we wanted to understand the relative information encoded within this neural substrate. One crude way of measuring this, we've thought about various ways over the years, and we have a couple of papers under review, but at this point, we just used Shannon entropy as a measure, the unique individual states of information in an electrode at any given time throughout the time series. We can see relative to normal appearing glioma infiltrated cortex had a lower entropy throughout the entire temporal series. Then again, as I mentioned, we can take normal versus tumor electrodes, and we can assign versus poly versus monosyllabic words. Again, polysyllabic words should have an increased high gamma power per trial per electrode relative to monosyllabic words. And while we can see that from a normal appearing electrode for normal appearing cortex, we cannot see that from cancer infiltrated cortex, right? You can't overcome that cortex cannot increase the computational demand required for a more complicated task. Similar sort of thing, we trained a series of classifiers to decode word condition. This is, again, using both frequency and syllables. And again, you can see where normal appearing cortex better than chance, we can train a classifier. These early classifiers we produced in 2021 were not that great, but they did perform better than chance. We've tuned this quite better over the years. But in glioma or cancer infiltrated cortex, classifier performance is 50-50. It's like flipping a coin, right? So this is not, well, this cortex is hyperexcitable. Cancer infiltrated cortex would not be suitable for many of the computations or more complicated computations we would need for a host to be able to perform, particularly in the setting of IDH wild type tumors. So the next phase of this talk was really to talk about any molecular drivers we could identify. Again, this idea that cancer doesn't just kill things, but remodels it electrophysiologically made us think, well, what are the drivers behind this? And so we have a couple of different ways of measuring tumor intrinsic connectivity or tumor-associated neuronal activity in the human. And one of them is more hemispheric and the other one's more local. And so our high-density subdural array is more local activity. The rest of this talk I'll focus on our more hemispheric connectivity, and that's using magnetic encephalography. And we assigned within the hemisphere, using contralateral hemisphere as control, in collaboration with Sri Nagajaran's lab, who has run this incredible MEG lab for many years. We assign by 8 millimeter voxels connectivity plots for each voxel within and around the tumor, again, using the contralateral hemisphere as control. And so we identified that tumor associated or tumor intrinsic within the region of flare, electrodes that have an upper tertile of connectivity, which we'll call HFC, relative to the lower tertile of connectivity, which we call LFC. By sampling these tissues within each of these contexts using bulk and single cell RNA sequencing, we were able to identify cellular populations. For the purposes of today, just to streamline this into a 25-minute talk, we'll avoid the myeloid populations, but we did identify that certain populations of glioma cells had various categories of synaptogenic factors, axon guidance genes, additional interesting gene ontology sets that were quite exciting. And so we focused in this paper on thrombospondin 1 because of its known relevance in astrocyte biology and astrocyte behavior. And we said this population of glioma cells within HFC or highly connected regions that are astrocyte-like and express thrombospondin 1 became our target. And we can see in these HFC regions, they have increased pre and post-synaptic puncta relative to the LFC or poorly connected intratumor regions. We put these cells into organoids. These are iPSC-derived induced neuron organoids. They don't form synapse-type structures. These are relatively immature neurons, but they secrete all the things that excitatory and inhibitory neurons secrete for the most part that we could identify. You can see, again, our HFC-like cells really loved and were trophic to our green neurons, iPSC-derived neuron organoids, cerebral organoids, where LFC, in our LFC condition, these cells seem to not care at all. We then again section these organoids, and we identified that again, if you take our LFC conditions, the primary patient-derived glioma cells, overexpress thrombospondin 1, we can make our LFC plus thrombospondin 1 cells behave in a very similar way to our HFC conditioned cells. Then we started to co-culture, and again, these are murine-derived neurons co-cultured with HFC versus LFC cells. We can see within 24 hours of co-culture, they become hyperexcitable. And another 24 hours, again, after 48 hours, they become not only that, but hypersynchronous as well. So we get this hyperexcitability, hypersynchrony when co-culturing HFC or highly connected primary patient-derived glioblastoma cells relative to those that are not. Again, inspired by the work of everyone in this webinar, we again decided in our HFC conditions, look at proliferation index. We can see that these are hyperproliferative regions of tumor, hyperinvasive. And you're running these in an EDU assay. We can see the HFC cells basically do not proliferate without the trophic factors of neurons. They really require the trophic secreted factors of neurons in order to proliferate, where again, our LFC conditioned cells don't really care. Frank Winkler's beautiful work really made us think about tumor microtubes and what's the effect of tumor microtubes in this condition as well. You can see this HFC plus conditioned media from neurons makes these cells form tumor microtubes, again, where LFC do not. Then we wanted to understand whether there's a survival advantage or disadvantage associated with this. And so we took our primary patient-derived HFC and LFC cells and xenografted those into the C1 region of a mouse. You can see they form these synapse-type structures that are electrophysiologically active at a greater rate, both in glioma to neuron and neuron to neuron synapses, in that these HFC xenografted cells have an increased tumor burden and greater proliferation index, and they kill these mice early. This idea that maybe thrombospondin could be pharmacologically targeted, we thought about that from a gene editing standpoint, as well as pharmacologic inhibition standpoint. Gabapentinoids are potent thrombospondin 1 inhibitors. Shala Eroglu and others have suggested this or shown this over the years. So we took our first approach, which is taking our co-cultured murine cortical mouse neuron, HFC plus or minus gabapentin, and looked at the reduction in hyperexcitability and hypersynchrony. Then looked at the proliferation index reduced in both these conditions, both with pharmacologic and gene editing. I'll say here that, again, after 1 month of xenografting, we then again sacrificed those mice after treating with another 8 weeks of gabapentinoids and looked at mouse survival, which was an advantage. We then turned back to patients and decided that we could measure serum thrombospondin in our patients based on the percentage or voxels of magnetic encephalography, imaginary coherence defined connectivity in patients. This is serum thrombospondin 1 in patients. That was quite encouraging as an early thought about potential biomarkers. I'd like to pause or caution us for a moment because, again, this was an important experiment that was asked during the review of this paper and comes up a lot. I see patients in clinic today that will ask, well, can I just be treated with gabapentin? Is that going to make my tumor shrink away? I think it's important to talk about these neuroactive drugs that do not think are potent monotherapies for glioblastoma. Particularly, they're not, if you give, again, without co-culture, you're not inhibiting or reducing proliferation if you give gabapentin alone. Really, it's activity-dependent proliferation that's impacted, more so than the proliferation at all. So we've defined these molecularly distinct HFC region cells that proliferate only in response to neuronal trophic signals, and they exhibit increased invasion and proliferation. They extend tumor microtubes, and they form synapse-like structures when xenografted. As I mentioned, what does this mean for patients? And we've thought about this a couple of different ways. First and foremost, I like the interactive effects of clinical variables on patients, not just the Kaplan-Meier survival curves. I'll actually show these different. I'll show this slide first. So the first thing we did, outside of just running a Kaplan-Meier survival curve for patients here that have HFC voxels within their tumor versus those who do not, that's the bottom left here. Blue, again, are those individual patients that have any defined HFC regions within the confines of their tumor versus those who do not. And we can see that these functional connectivity voxels within the tumor for patients is a survival disadvantage for this population. We equally matched by everything that we could understand or see from our patients, but we decided to run a similar analysis looking at a population of patients, again, purely receiving all standard of care temozolomide treatment, temozolomide and radiation, or the STOOP protocol. And then we decided to say, okay, what are the major prognosticators of how long these patients live to the left? Using recursive partitioning, we identified that, again, as you might imagine, age and extended resection are important variables on survival outcome for patients with glioblastoma. But it turned out that is HFC or tumor voxels that have intratumoral connectivity was a strong prognosticator, again, equal to the effect or similar to the effect of age for our patients, and really defined risk group 2 in terms of our prognostication. So this presence of connectivity is not just therapeutically relevant, but also may be prognostically important. We start to talk about clinical trial applications. I think that's desperately what our field needs. And so as we started to talk about that, we went through the NRG, the NCI-backed clinical trials consortium, and they started to ask about how we could make a neuroactive drugs, or particularly gabapentinoid class, useful as a clinical trial concept. And we said, well, yes, what evidence do we have besides our preclinical evidence? So we looked at a couple of annotated data sets, one at University of California, San Francisco, and the other in the Mass General Brigham system, and used a couple of machine learning approaches, as well as annotation of that data set. And long story short, that you can see, again, if we look at the Kaplan-Meier survival curves, in both of these data sets, the use of a gabapentinoid drug is a survival advantage over those not. The average drug for those patients treated was about 450 milligrams. And for those patients in these systems that have had blood or serum available that was biobanked, we can see again that the use of that gabapentin did result in a reduction of serum thrombospondin in these patients. Again, these are crude early experiments. This was published earlier this year as helping us to get the evidence or to lead up to a clinical trial. But this has really been one of the reasons that this trial has been greenlit, or has been tentatively greenlit by the NRG as a window of opportunity study for the next stage. So again, moving beyond this idea that these are they're important for the biology of patients, biology of, again, malignant brain and IDH wild-type gliomas, important for their biology. We think it's prognostically important as well. Gabapentinoid drugs and tumor intrinsic connectivity and measures of neuronal activity, we think are very important, again, predictors of outcome and therapeutically relevant. And I hope we'll have more and more trials over the upcoming years and think about how to intelligently combine these drugs together so that we can improve the lives of our patients. So just a quick point, take-home points. I think glioblastoma or diffuse gliomas are really more than ablative diseases. They really remodel circuits more than just ablate neuronal populations. I will not show the data, but we actually have really robust data that's going under review right now about neuronal populations that are vulnerable to the presence of diffuse glioma versus those that are more resilient, and why we think that is. Task-specific responses are maintained. And that's important as we think about therapeutics and cognitive outcomes for patients. Tumor intrinsic connectivity, again, is a prognostic disadvantage for patients. And it can think as we try to identify and target these mechanisms of connectivity to improve survival, we have a number of potential targets that we can begin to test in the clinic. So this is my lab and all the people who really do the work and our funding sources. I want to thank everyone for giving me the opportunity to hang out with you all for this webinar. This has been wonderful to watch the field emerge, and I hope we have a few minutes for questions as we all come back together. Thanks so much.
Wonderful talk. Thank you so much, Dr. Hervey Jumper. I want to make sure that everybody has a chance to ask questions, but I have one that I've probably asked you before, but it keeps coming up whenever I hear this, you present these beautiful data. This really prominent language circuit remodeling is so striking, and it occurs over such a large geographic area. And I think, thinking about changes in synaptogenesis, clearly that explains some of it, but some of the distance that needs to be traveled would suggest there needs to be additional structural, perhaps, mechanisms like axonogenesis or axon sprouting. Have you looked into that? Have you seen any evidence of axon remodeling induced by tumors?
Am I still? Yeah. That is so fun. Actually, yes. And I just took my first grad student for that project. I think, yeah, axons are tricky. These are tricky experiments. And I wanted to make sure we understood the basic neural circuitry before we talk about axon structures and white matter, right? And I said that mostly because in my interpretation of the literature, what happens in subcortical structures under normal conditions is actually much less understood than even cortical structures. And I wanted to have as strong as an understanding as we could on the cortical neuron circuitry, but there are certainly molecular drivers that suggest axonal differences in myelination differences. And some of, Michelle, your early work that when I first met you was really building the framework for that. So the papers we've been looking at to think about these experiments in humans are actually informed by I think it was a 2015 paper, if I remember correctly, that set the stage for it. So you're absolutely right. It's very preliminary. We've run our very first couple set of experiments now, and much more work to do in that space.
Great, thank you.
And there are ways. There are
sorry,
also
go
ways
ahead.
of measuring this. CCEP, like cortical evoked potentials, and we have human assays to run these experiments as well. So it's it's a good time to ask these questions even in people. Sorry.
Wonderful. Thank you so much. Dr. Sontheimer has a question.
Yeah, I'm really intrigued with your gabapentin study. And one of the questions I have here is, since this is a retrospective rather than a prospective analysis, is it possible that the presence or absence of seizures in these patients has guided whether or not they receive gabapentin or not? And how can one separate the therapeutic benefit based on them having gabapentin or them actually having a disease that presents with, with hyperexcitability and seizures versus not?
Yeah, thank you so much for asking that question. You ever write a paper and you're almost apologetic, like, I know this confound's here. I can't control for all the confounds. That project was only written because after about a year of working through the clinical trial concept, we had a massive data. Said, all right, let's go ahead and publish this. But that's the whole reason that we need a well-controlled prospective clinical trial in folks, documented without epilepsy. None of these patients, according to the medical records, had any more than a single presentation seizure, but we can't confirm that, right? And in 500, 600 plus patients. So these are, again, retrospective data sets that are annotated and controlled to the best we can, but the only way we can confirm that is the prospective study.
It'll be real important. I mean, I learned from my clinical colleagues that there's different philosophies as to whether or not to put glioma patients on prophylactic anti-epileptics or not. And some subscribe to it and others don't. And so I guess that would explain why quite a few of your patients probably are on gabapentin without them actually having epilepsy.
So I will share as a practicing neuro-oncologist, gabapentin is a very weak anti-seizure medicine. It's not at all one that we reach for seizures. However, it is a very good medicine for neuropathic pain. And so people are on gabapentin because of neuropathies or because of, other, if there's a thalamic involvement of a tumor that causes a thalamic pain syndrome, we may reach for gabapentin. But the vast majority of people who present with seizures are put on most usually levetiracetam because it's metabolized by the kidneys rather than the liver and doesn't interfere with other medicines they may be receiving for their cancer. So I don't think that presentation of seizures are likely to be a reason that patients were on gabapentin, but other pain syndromes, low back pain, et cetera, is more likely.
That's exactly right. These are almost everyone was on it because of chronic pain. That's exactly right. Beautifully put. But they were on other anti-epileptics.
Yeah.
Just not that one. And then with trial design became the problem of, okay, patient has chronic pain. What do you put them on that doesn't interfere with the trial? That's actually been several months of conversations about what would be the appropriate neuromodulatory pain medicine that these trial patients could be on. Right.
That doesn't also affect neuron glioma interactions.
Correct.
Turns out a lot of them do.
You can see why it's been
complex
months
as we start to understand more.
It's been months of conversations because of that. Yeah, we're not exactly sure yet.
Okay, let me know what you guys decide on.
Yeah, if you have I might loop you back in. If you were a part of that in the beginning, but
yeah, lidocaine, but it's not. Lidocaine patch. It's tough. It's really tough.
Yeah.
Wonderful. Any other questions specifically for Dr. Hurry Jumper, or should we move to now the sort of group discussion phase of things?
Group discussion sounds great.
Okay. It's so exciting to see these, really consistent themes emerging from, your independent laboratories. The way that gliomas influence the nervous system and the way that cancers in general, influence the nervous system, and particularly, neural circuits and brain functions. I think we're really just at the tip of the iceberg in understanding. So let me ask the group, what do you think are some of the biggest open questions?
I'll start. So as you and I have spoken about, it's not a one size fits all, right? And that you can imagine a combinatorial code of different types of brain tumors, different types of circuits, differentially influencing brain tumor growth. And I think what's really important is sort of as we move forward as a field is decoding circuit-specific relationships between tumors and neurons, because we, as Dr. Sontheimer alluded to, location matters, right? The type of tumor, like the genetic makeup, is going to influence things, and so I think really getting a little bit more granular. I mean, it's a natural step in the field, right? And I think we need to get a little bit more granular and decode some of these relationships with different types of neurotransmitters, different types of receptors, locations, and tumor types. That's from a basic science perspective, right?
Yeah, absolutely.
This grand scale mapping, I think, which is certainly can be extremely revealing and also guide us which cancer subtype, what is the most promising, let's say, intervention. So this is very hard for a single lab to do, right? We have ideas kind of to do this in a consortial way, or how to kind of contribute this in a meaningful way that in the end we end up with really a meaningful panopticum of this of these interactions. Ben?
Consortium sounds good. No, you're right though.
Particularly a funded consortium, that would be wonderful.
Exactly, yeah, funded would be better. Yeah, no, but I think Frank's right though. It has to be from around the world, right?
Yeah.
And this is task, right?
Yeah.
If everyone picks a tumor, everyone picks a circuit. There you go, right?
I think the field pretty quickly we need to, clearly everyone is here because we think it's not just fun biology, but relevant for people. It matters, right? If we're taking, if we're guided by some of the recent successes in the cancer world, having very precise endpoints of what therapies should look like, I think that's what's needed, right? We have treatments for IDH mutant gliomas because we had beautiful endpoints. We know what success looked like, right? And so I think we need that sort of thing, electrophysiologic endpoints that are letting us know as we cycle through the various drugs. Otherwise, we'll have a few, clinical trials. We'll have a few drugs that may or may not show effect on 6-month progression-free survival. And then it'll, kind of go away, right? If we don't know what we're measuring. So I really hope we get these endpoints pretty soon.
Yeah. No, I agree. And I think as we begin to design trials, we really need to understand the tumor-specific biology, as Ben said. We found, for example, there are really profound effects of, GABAergic neuronal activity in pediatric, diffuse midline gliomas, not present in, adult IDH wild type hemispheric high-grade gliomas. Just as one example, if we design a trial that targets some of those interactions, we need to make sure we're measuring things in the right tumor and also to anticipate what to see with respect to endpoints. Do we expect to see single agent efficacy? What does that look like? And more likely, what's the best treatment regime, to include a neuroscience inspired therapy in, and then how do we measure the difference? Harold, I know you have your hand up. Please just jump in, but please.
I'm going to pose what I actually heard throughout all these talks and throughout all your published work. I think of a stronger focus on understanding what happens to astrocytes in the context of tumors, because clearly, as an astrocyte biologist all my life, I've been really fascinated just to how important astrocytes are in synapse development, in circuit development, in homeostasis, in sealing off lesions, in attracting immune cells, in presenting antigens. There's just so many functions, and I can't fathom that these tumors grow next to an intact astrocyte. So they have to reprogram and hijack astrocytes, and that might be a tremendous opportunity by understanding, coming up with astrocyte targeted therapy. If you can keep the astrocyte network healthy, you might actually be able to slow the glioma network.
Yeah.
I can't tell you how to do it, but I think that certainly I find an exciting area.
Yeah, I totally agree. And outside of the people on this call, there are a few others, but there's been very little work on tumor-associated astrocytes, and furthermore on the effects of these diffusely infiltrative, kind of brain-wide, distributed tumors on the astrocyte network, how that regulates excitability, how the astrocytes are promoting growth and limiting anti-tumor immunity, I think are really important questions. Agree.
We need to really understand the astrocyte subtypes involved, right? Which now kind of our knowledge about more and more astrocytic subtypes now, which are really operational in glioma and to what effect. We are seeing that these, and this is kind of largely unpublished, these glioblastoma cells, when they send these long membrane tubes interconnecting to these communicating network, they actually do the same with astrocytes in the tumor environment. So there's a massive calcium wave exchange and electrical exchange and calcium wave communication between the glioblastoma cells and other astrocytoma cells also with the neighboring astrocytes. And then in this, while we all kind of have found that the neuron glioma synapse is more neuron to cancer, this is other way around. So we see that the cancer cell is largely generating these activity, and then the astrocyte takes over. This must heavily influence astrocyte biology in the tumor microenvironment, also potentially contributing to seizures, and because we know astrocyte, as you said, very right, kind of largely involved in seizure generation, etc. So we have really a situation where these cancers are heavily integrated into various cell types of the normal brain, and this definitely will change the kind of normal functions. And this is a complex system, so we really need to study this we can't study this in the simple in vitro cultures. We really need to kind of, it's getting a bit more complicated, but it's also quite exciting. And in the end, by integrating into these astrocyte networks, these tumor cells, of course, even gain more resistance and have a large communicating syncytium, so to say, with normal astrocytes. And yeah, I think that's certainly, astrocytes are I agree, an important hot future topic for cancer neuroscience in the brain. Yeah. And also particularly what Ben is doing, right? Ben has some very, I would say, crazy papers in the pipeline regarding the function of astrocytes and memory formation, right?
And engrams, right? Yeah.
Right.
We're doing stuff there. We have a few thoughts, but to Dr. Sontheimer's point though, I actually think neurons are easier to study in this context because there's more tools and we know more about them. It's actually lower hanging fruit. As Dr. Sontheimer knows, studying astrocytes is really hard. We don't really know what they do, and the tools that we use are really just adopted from the neuron side of things. Mm-hmm. So that's a real challenge, and it seems like the astrocyte is the stepchild of just about everything. And so applying that biology to this is going to be a challenge because we don't have the tools and we don't know what they do. And there's a lot of mess in the field with reactive states, whatever that means. And so I think it's a great idea. We have to do this, but I think we need more basal foundational knowledge before we can really dig into it. That's why I went on the neuroscience, because there's more knowledge there. It's a little bit easier, in my opinion. It's all really hard. It's just a little bit easier.
For the future, nevertheless, right?
Yeah.
Now, as I was listening to your talks and again integrating that with Frank and Michelle's work, I was thinking if remodeling of circuits that involve GABA, glutamate, serotonin, gap junctions is so instrumental in tumor biology, would it be a crazy thought to induce a coma and to literally quiet the brain for 2 weeks or a period of time to disrupt these networks from forming? And has anyone done that either in, I could see this might have been done in the 1950s in humans, or has anyone done that in animal models? And this is a crazy idea.
So it's not, been studied prospectively in clinical trials. And I think that the issue is that it's not 2 weeks you would need to induce a medically induced coma. It's a very long period of time during which you need to effectively remove the tumor cells. And that's also a big challenge. So it hasn't been done. I think what I tell patients, because this idea has, this question has been asked, is, our job is not to impair your ability to use your brain. Our job is to find the molecular targets that disrupt the cancer's ability to take advantage of it. And I think for patient quality of life and the myriad medical complications that something like that, brings up for a patient, from deep venous thrombosis risk to, infections and many, many other very, very serious risks, that disrupting the specific interactions, I think, is a much more medically feasible approach. But it, certainly has come up in discussion, and it, it is true that when anecdotally we see a part of the patient's nervous system become disconnected from the rest of the brain, tumors tend to stagnate there. And it's really clear in optic pathway gliomas. When an optic pathway glioma progresses to the point that the patient loses vision function, the tumor stops growing.
So do hibernating animals get brain tumors?
Do what? Hibernating animals? Well, sleep is not coma, right? And I think this is important. There's a lot of activity during sleep. It's just a different pattern. It's just a different brain state.
So when we apply coma in mice and do the intravital two-photon microscopy and then compare with awake phases after that, we can make invasive cells stop invading during the artificial coma. We can stop the entire system. There's no calcium activity anymore or very reduced. So yes, it does work, but unfortunately, as Michelle said, it's unfortunately not really translationally feasible. We are studying now how sleep, which is of course no coma, but a very different state of mind, a very different state of brain, how sleep influences all this compared to wakeness. And I'm really looking forward to this data. Yeah.
Well, to piggyback off of this, there are frequency changes that are perturbed and are pitched as cancer-directed therapies, right? If you look at the new product insert for Optune, that's what they're describing. They're describing a beta frequency oscillation change that therefore, right, and therefore their clinical trial. This has been more post hoc than not quite how it was described pre, but there are companies now that are building devices. One is an oral device and others are head mounted that are describing changing frequencies over long periods of time that's non-invasive in a way to treat brain cancer.
And this, leveraging the circuit knowledge and leveraging the incredible advances in functional neurosurgery, this is a very promising approach to treat, the circuit biology, the circuit pathophysiology of these tumors and slow them down, combined with some effective strategy to remove or kill the tumor cells. I think it's a, very, very promising approach. So not fully shutting off a person's brain function, but modulating it in a therapeutic way.
I think we have so many clever ways of neuromodulation, and which are also kind of even used in neurorehabilitation to create effects. So I think we can probably do even better effects than the electrical fields. But we'll see. We at least have seen that the alternating electrical fields are heavily influencing this tumor microtube cancer. Calcium communication in vitro monoculture, etc. So let's see. I think it's particularly important to see what these alternating electric fields are doing to the real mouse, and we're trying to set this up also at the moment. So that will be interesting, because I think we really need to jointly develop, particularly with our neurosurgical colleagues, really ways of how to interrogate the operation sites, so to say, the operation theater scene, to modulate these networks with different, be it by electromagnetic or direct electrical or whatever means, to see how we can optimally influence these networks.
At the very least, it might be nice to understand how some of the behaviors that people do influences cancer growth, right? That becomes a question all the time. So patients engage in various types of meditation acts and other things, and always have questions about, if activity matters, and does the way I live my life influence cancer growth? And Michelle, as you know, your paper opened up the floodgates to that sort of line of thinking that patients and families have for clinicians. So at the very least, we need to understand that better.
And in particular for brain tumors, right? Because people talk about like other forms of, but really in the brain, right? How brain states influence brain tumor growth, I think is also, just a general state, not a circuit specific effect, right? That's something to think about.
Yeah, no, I agree. And we have lots of intriguing clinical observations, along those lines, but you really need to understand the underlying mechanisms.
I have a really full handful of glioblastoma patients who shouldn't be alive today, but will live for 5, 6, 7 years. And they all told me from the beginning, you know what, I'm not afraid of this disease. I will do my meditation, my yoga, my whatever, and I will control the disease. And they were kind of extremely self-confident. I always said in the beginning, yes, if you think it's good for you, do it, whatever. Yeah. But now I really it's really striking me that these patients still live. and they stood out in the beginning. So I gave all of them homework now when they see me in my outpatient clinic and say, okay, please write down exactly what have you done. And so I get a kind of a half, I'm doing half control interviews with them now to trying to figure out, maybe we should really combine these experiences in our sites and try to come up with a at least working hypothesis that might be even be then tested in the prospective clinical trial, which would be fantastic, a fantastic thing to do. I would love to participate there and work on that.
Yeah. Yeah, and this brings up, the bigger questions again to this idea of brain states. Are there pathogenic brain states induced by the cancer, brain tumors or other tumors elsewhere in the body that, not only deleteriously affect the patient and their quality of life, but also encourage, and allow for cancer growth through complex brain-body connections. There's just a lot to learn.
We know that stress and stress hormones and pain and anxiety can drive extracranial cancers, right? As shown by Elizabeth Rapasky and many others. So I think there's so much to learn how our mood, our emotions, our brain states influence cancer in the brain and throughout the body. I think that's really amazing.
Yeah. And how cancer affects mood, right? We were taught that patients with new onset major depression and a little jaundice have pancreatic cancer unless proven otherwise, right? This is how 80% of patients with pancreatic cancer present with a major depressive episode for the first time. So there's some really complex things happening between the nervous system and cancer, affecting brain states.
Yeah.
Much to learn.
Yeah, a lot of arguments for a vicious cycle on a really organismal level here, right?
Yes, absolutely.
Body to brain communication.
Absolutely. But we're off to a good start in the field, starting to understand how cancer influences the brain, in large part thanks to the work of these three fabulous speakers. So thank you so much for these wonderful talks and for your beautiful work, and really excited to see where the field takes us.
Well, thank you for having us.
Thank you, Frank and Michelle.
Yeah.
Thank you for being with us. This was a wonderful seminar. Thank you so much.